All right, I see I guess we've lost a bit of resolution, but my name's John Lawrence, and I'm here to give you a little bit of a talk about about biofilms, and the title is How Microbial Slime Can Change the Quality of Drinking Water. And certainly biofilms and associated slimes can certainly have negative effects, as you've seen in water treatment and sewage treatment and so forth. They can certainly have very beneficial effects as well. I guess we're going to talk mainly or mostly about some of the negative effects. Now, they can form just about everywhere because certainly microorganisms are just about everywhere, kilometers deep in the earth, miles high in the sky, and on all surfaces and so forth. So it's very easy to introduce microorganisms into any system, even if they're not there already. Now, the biofilm itself certainly has organic and inorganic components, as we can see, and it uses both sorbed and soluble nutrients to grow. And you can see that there's certainly a tremendous potential for biofilm formation in drinking water systems. You have a very nice surface, you have lots of moisture, you have the potential for nutrients, and I'm sure you've heard about the necessity of controlling nutrients and carbon sources going into drinking water systems in order to reduce available nutrients for biofilm development. Now, what is a biofilm? And there are a lot of complicated definitions for biofilms. A community of microorganisms attached to a solid surface, including bacteria, fungi, yeasts, protozoa, and other microorganisms, or most simply the slimy layer of microorganisms that covers solid surfaces. And if you haven't brushed your teeth for a while, or if you've slipped on a stone in a stream, or any one of a number of other issues, if you've had certain types of infections, you've had experiences with biofilms in a fairly intimate way. If we look at simple examples here, hot springs, streams, and rivers all have biofilms in abundance. They're medical, you have dental plaque, various types of infections associated with devices or catheters and things like this, agricultural on plant surfaces, and industrial, which we're related to most here, heat exchangers, filtration units, wastewater treatment, where, as we've said, it's quite a good thing, ships' hulls, where it causes tremendous drag and a lot of energy loss, and of course drinking water systems, where we'll have a little look today at where biofilms may develop in a number of different places. Now, I just want to show you, give you a little bit of an idea of scale here. And this is a thumb and finger, and this is a pin. So we're not talking about angels on the head of a pin, but we're talking about bacteria on the head of a pin. And if we want to see what we're actually looking at with a lot of our micrographs and photographs that we'll be looking at, we can just go in here and have a look at this pin and see here. And in general, a lot of the things that I'm talking about actually take place in an area about that big. So two of these squares here is about the width of a human hair. So a lot of these processes take place on a very small scale with very small organisms. And this is just a computer rendering of a biofilm that we've done and stuck on the head of a pin to give you a little bit of an idea of what we're looking at. Now, here we're looking at that area, only this is on a spinach leaf, and this is a leaf just purchased in a store. And you can see here, these are plant cells, the red area. All these little green things here are bacteria growing on the surface of that leaf. So if you've got some wilted lettuce that you've bought and it seems a little slimy, you've got a biofilm growing on it. In general, that's not really a big problem for you, although in Vancouver we did have an incidence with Shigella as a result of spinach that was improperly washed and packaged, and so pathogens can be associated with these biofilms. Now, they can also be involved in mineralization processes. And here up in the Yukon, coming out of the side of the hill, there's a little spring here, and you see all this yellow stuff and this little rilled structure, and this is iron that's coming out, and it's precipitating on a biofilm. And the biofilm is made up of algae and bacteria. We can see here again, going back to that little head of the pin, these are the cells that are growing in that yellow material. They're surrounded by a bunch of goo, polymeric materials, and that material precipitates or absorbs iron and other minerals and causes this concentration of materials around these cells of these microorganisms. So you can get a very large deposit like that as a result of water, groundwater coming out, running over organisms and then absorbing and mineralizing. Let's just go back to that one. So here's the macroscopic look at what you see, and here's the microscopic look showing the cells here surrounded by polymer and all sorts of minerals and stuff embedded in that polymer. And this is a process that's quite important in terms of biofilms on a number of scales, but can be quite important in terms of drinking water systems as well. Now, if we look at water system biofilms, we can see here a little plastic strip about 1.5 inches long, and it's covered by all this goo, and there's quite a bit of goo here on the end of this thing. This is a river biofilm that's quite a bit thinner. This is up to about 4 millimeters thick, and it just has extra carbon applied to it. This biofilm here is ordinary river water being pumped over this surface, and you get a fairly thin biofilm that's maybe 4 to 5 human hairs thick in terms of its structure. Now, I just want to show you an image here. Again, this is the head of a pin area, and this is going to be going with a microscope, a laser microscope, and focusing through the biofilm and showing you a number of different things that are present within a biofilm in different layers. So, okay, so we're focusing through that. Then this is the bacteria. These are the algae and so on that are part of that biofilm. And then this is putting all of that together. And you can see all of these different parts with algal filaments, bacterial cells, and polymer, all that slimy stuff built up and developing. And this is, again, an area of biofilm that's about that could fit easily on the size of the head of a pin, but obviously we have thousands and thousands of different bacteria, algae, and various organisms present within this biofilm. So it's a complex community of organisms. We can look at it here in a river biofilm. This color is false color that we've introduced, but you can see here very many different types of bacteria present. Some of them may be pathogens, some of them may be opportunistic pathogens. Most of them are just bacteria making a living together in a community, doing important things such as processing carbon, cycling nutrients, and a lot of things that are very important in terms of the functioning of natural ecosystems and the functioning of wastewater treatment and a lot of other systems. Now, to look at biofilm development, I thought we should sort of step back a little bit and see exactly what happens in a biofilm. And this is a little cartoon series, and I'll caution you that bacteria generally don't wear sunglasses and are not to be found sitting around in lawn chairs, etc. But a colleague of mine, Sonia Seftek, who's also works on biofilms but really has a love of cartooning and animation, has created this little explanation of what happens in a biofilm and how they form. So generally, biofilms develop because nobody likes to live alone. Although traditionally we've studied bacteria alone. They like to live in crowds. So you have a surface here. You have all these different bacteria going by, and you get organic deposits on the surface and other material developing here. And eventually it becomes attractive enough for microorganisms to settle down, and that's just what this guy's done. And bacteria tend to attract each other to the surface, and eventually they set up homes, and they multiply, and they develop into a biofilm. So that's a very straightforward expression of how a biofilm actually comes to be. Now, it's a little more complicated than that, and the bacteria, although I find them to be very attractive when I look at them in a microscope, they don't generally smile back or have little eyes that look at you. But that's basically what happens, is that the biofilm develops because microorganisms find a suitable surface, it's enriched with a bit of nutrient, and they attach and they grow there. So if we look at this, well, I guess we'll look at it again. So you can see here, this is basically historically, I guess, from some people's perspective, an error in microbiology. We deviated from studying the communities of microorganisms and really got into studying only individual organisms. And only in the last 20 years have we really gotten back to looking at communities and seeing how they settle down and how they actually chemically talk to each other and attract one another, and how they produce all of these exopolymers, and how they grow as communities and families, which are referred to generally as biofilms. Looking at it a little more schematically, we see the bacteria going by, we see the surface becoming modified by all of the material that's in water, bacteria attaching, forming little colonies. The colonies coalesce, they produce polymer, and then eventually it gets thicker and thicker, and you get all sorts of different bacteria present within that system. And this is just showing the same thing, only these are actual photographs of a biofilm development going on, developing from about 7 days old to 14 days to 21 days to about 30 days to 40 days to 60 days. So in about 60 days you can go from having just a few little things about the thickness of a human hair or less to something that is quite visible to the naked eye and would feel slippery to your thumb. And that's what develops on most surfaces that are not put into clean, clear, or pure water. So drinking water biofilms, the systems are important for biofilm formation, and they'll influence the supply, quality and quantity, and they'll also are issues with water safety where you have pathogens which can become parts of these communities. Every community has perhaps something objectionable about it, and biofilms can have members that are potential pathogens. And this is what we're going to actually have a look at. Now, how do these biofilms arise? Usually there's a few factors that are important. You get improper installation practices, inadequate flushing, back siphonage, faulty storage of water. I'll give you an example of improper installation. In Berlin, they built an office building. They pressure tested the copper water system in the building. There was a delay in finishing it. They didn't drain that out. They didn't properly flush the system. A biofilm developed on the inside of the copper pipe, which actually corroded and pitted the copper pipe to the point where there were pinhole leaks throughout the entire copper system in the building. When they put the water on, it was essentially a 14-story sprinkler system rather than being actually a water system. So they had to completely gut out the building and rebuild it because of a microbial or bacterial event within that water supply system. So one has to be careful. Construction repairs can introduce things. Large outbreaks usually are inadequate treatment or disinfection of the water source, but there is evidence for outbreaks as a result of system problems, distribution system problems. Now, looking at water wells in particular, we have an excellent site for biofilm growth, both inside the well, inside the pipes, and inside the porous media or the sand pack around that well. There's a lot of opportunities for biological plugging and water quality changes here. And we can see biofilm developing inside these pipes and biofilm developing in the sand pack around the well. And this causes sanitation problems, plugging problems, and water supply problems in a lot of areas. Now, there are a lot of wells in the 3 prairie provinces. If we look here, 1 to 6 wells per town site, greater than 108 wells per town site. It actually follows human habitation pretty well in terms of the distribution of wells, but wells are a very, very common source of water, particularly in Western Canada. Millions of people depend upon the water supply from groundwater in particular. And this represents about $1.3 billion worth of infrastructure that people really don't think much about, that has a very interesting story in terms of development. Now, what I'm going to show you here, if you look, this is about a 4-inch diameter pipe, And this is the outer edge, and what we've done is just put a camera through this pipe to take a picture of the biofilm that's developed here over a couple of years and see what that looks like. So here you can see the biofilm. Like, this was a nice clean pipe when it was installed, but you can see all this biofilm. Here's some material being disturbed and falling off the sides. So there's quite a bit of biofilm. You can see a lot of mineralization of the biofilm, iron deposits. Here there's actually ropey structures that are a little reminiscent of going to the Titanic, actually, growing inside the pipe itself. But you'd expect this, when it was installed, it was a nice clean pipe, but biological processes, nutrients in the water have all allowed the development of very nice biofilm on the inside of that pipe. So this gives you an impression of what the inside of your well or the inside of the pipeline coming to your treatment plant may look like after, in some communities, after only about 6 months. In other communities, it may take 3 to 4 years to actually get to that level of fouling. So you can see that you can get serious clogging here in this particular pipe. This is iron, and people recognize the iron, but there's a microbial component to that, the biofilm that is mineralized and actually plugs that up. And that presents a problem in terms of supply. And if we look at things, we can see here with this pipe, there's quite a nice biofilm developing on the inside of that. And here's a shot inside the pipeline. We see these nice ropey structures and a heavy biofilm on the inside of the pipe. And of course, on pumps and other sections, you get biofilm, mineralization, and things that will negatively affect water quality. So, now that's what happens inside the pipe. One of the things that interests us is what happens inside the sand, in a filter or in the sand around a well in particular, because there are problems with controlling this and developing it. So what we've set up here, these are sand grains in a sand pack. So we're looking into the sand using a laser microscope, and we've added a little bit of nutrient to the well water to speed up the process, so that rather than being here for days and years looking at what's going on, the events that we'll see in this little movie happening around these sand grains takes place over a few days, and we'll watch it in a few seconds. So you can see flow going by, and you can begin to see little tendrils and little fuzzy surface developing around these as the time goes by, about 19 hours here. Now there's a tendril developing on the surface of that. This is biofilm developing. Yeah, there's some sloughing of biofilm happening in the system. Big growth in here, some plugging happening up in these pore throats. And these are just bacteria that are developing, and you can see here we're at 2 days. There's a lot of material sloughing off of the biofilm and going through this pore space here between the sand grains. And if you watch this here, this is actually the back pressure in the system. So you can see the pressure building and releasing as a result of plugging. And a lot of sloughing, because biofilms eventually, they're fairly stable, but they'll release material as time goes by, and that's what can cause problems with pathogens and other things being released from the system. And here we're out about 4 days, and you can see the pressure really building up. There's still water getting through here, but this is almost completely plugged, and we have quite a high pressure across that material, and now after about 5 days it's completely plugged, and you're not going to be able to draw water through that material. And that over years around wells and in other materials that rely on biological processes, that's what you're fighting against, is that particular process. So we can see here, just looking at frames, the development of the biofilm, this white material on the outside of the grains growing with time. You see the flow, it narrowing off and eventually plugging up. And that's a process that is a bit of a bane in terms of water treatment and sewage treatment, in terms of recovering water from aquifers and so on. You get this biological plugging that can occur. And just a little bit more showing not only the biological plugging, but also gas bubbles being developed as a result of the biological activity of the biofilm. and just simply sealing off the system. So, why worry about this? Well, plugging and loss of yield, declines in water quality, increased microbial contamination, a greater likelihood of pathogens, and increased costs and potential risks are all associated with this biofouling process. And because, in particular, water wells and rural water distribution systems are often described of as set and forget, because it's installed, water's pumped out of it, and unless there's a problem, there really isn't a lot of maintenance or checking back done. Now, there are various solutions to the problem. You can pig out your pipes or your well. You can use pasteurization. Acid-base treatments have been tried. Electrochemical treatments is something that we've been working on over the last few years to see whether that can have any positive effect. There are a number of other antimicrobial agents that were referred to in the previous talk that can be used to try and clean up these kinds of problems. Here's a nice little selection of pigs. They may remind people of other things, but you just stick these in the pipe, blast it through, and we can see here a gentleman who's loading a pig into a pipe, and this will just be pushed through by water or air pressure, and it'll simply peel off the biofilm as best as possible and push out a lot of the material which will be collected downstream. Now, the problem is if you scrape a biofilm off, such as we've done here, and leave the natural, nice, thick, gooey biofilm over here, just as a comparison, you can see what happens there. Now, the thing is, here's our time 0. Here we've scraped it off. There's always somebody who survives scraping or pigging or anything. Within 21 days, we're pretty much back to where we were before. So unless you can completely eliminate that by applying residual chlorine or other techniques to try and suppress that, they're going to come back again fairly quickly. You can try acid-base treatments and then pump the well out or pump the line out. You can see the water coming out here is bright red with iron and high bacterial counts. And after about 2 hours of flushing, eventually you're back to having fairly clear water coming out of there. So the treatment works for a period of time. But if we look at the sort of history of a Saskatchewan water well here, we can see here this is where it's drilled. And it produces about 20 gallons a minute, which is actually fairly good. And it goes along for a period of time, a period of years. This is 95, this is spring 2004. And you can see you get a number of years where it remains at that level, and then it begins to drop off in here. And you can see there's a loss of yield. And with wells have different sort of decay rates. And sometimes it's fairly quick. There are some towns in Saskatchewan where the water wells basically last a year or 2 years before you have serious problems with yield and water quality. So interventions can be done, and here acid-base treatments have been applied, and you can see it recovers quite nicely, but then it gets right back onto its decay curve. And then you can come in and scrape the well out and do an acid treatment, and it recovers a bit, and then it goes down and gets back onto the line of decay that it was on. And then you can clean it and do a mechanical thing. And then you can even apply electrical current, and it'll improve it a little bit, but then it'll go down. So you increase the electrical current, but then it continues back down again. So you're always fighting a biological process, which is living and active and going to always fight back and get back onto that line. And so here you've spent maybe $12,000 on this. You've spent about $5,000 or $7,000 on this. You've spent about $10,000 installing that. And you originally spent about $50,000 to $60,000 installing your well. So at each point, you're judging your cost against the cost of having to replace that well entirely. And these are not unsubstantial. So there's a considerable amount of interest in doing something that would change the slope of that line, or that would permanently recover that well to a level where it would produce what you need to supply your town. So, why are biofilms such a hard target? There are a number of things that are a hard target in the world, but biofilms are in particular quite resistant. They have special metabolic states. They can just absorb the antimicrobial agent or the toxic compound you put in to kill them. They respond to stress in special ways by lowering their metabolism. They just don't do anything for a while. There are special cells that can persist in very, very stressful environments, which means that they can stick around and survive all that chlorine you put in, or all of that chloramine, or all of the other things that you may put in to try and eliminate them from the system. So if we have a look at this is just showing a biofilm with penetration of the biofilm by a fluorescent compound, which simulates the material that you'd be putting in to try to kill the biofilm. And we can see over the course of about 2 minutes, there are bacteria everywhere here, but this just doesn't penetrate everywhere. So the bacteria that are in here, they don't even see that compound that comes in. So as a result, they survive that event. And we can see here that, again, think back to looking at the head of a pin, that there are a lot of different areas within a biofilm that have different qualities and habitat and will respond very differently to antimicrobial agents. So they're just heterogeneous enough to survive almost anything. So if we look at this in a schematic sense, if you add the antimicrobial compound, the chlorine or whatever, it doesn't penetrate evenly. It may be absorbed, it may be broken down, and the inner layers may have time to initiate a response to this, where they basically shut down and wait till conditions get better, and you have organisms that can persist. So you get incomplete eradication of the biofilm with organisms available to regrow in that system. Now, the other problem that we have is harboring pathogens. We've now detected Helicobacter, which causes stomach ulcers and other problems in human beings in Mexico City water, in various UK water systems. Legionella, which causes a type of pneumonia, is very common in water supply systems and can be isolated and shown to be present. E. coli is certainly present in water distribution systems around the world and can survive in biofilms for extended periods of time. In fact, these pathogens can establish special relationships with organisms within biofilms, which allow them to persist and allow them to survive, and then be periodically released into the system, presenting a problem. So we saw a lot of sloughing in that little movie in the pore spaces, but you get a nice thick biofilm with pores and channels in it, and then you'll get sloughing with material coming off, or you'll even get cells that periodically just leave of their own accord. So you get little bits of biofilm and individual cells released into the bulk water in the pipe or in the well system. So these will appear periodically and cause some problems. Another method by which their pathogens are harbored in biofilms, pathogens can survive within protozoans. And this is certainly true for Legionella. So you have little amoeba and other things that can live within this biofilm community. And they can certainly do this for Legionella, and they can also do this for some chlamydia-like organisms as well. And if we just look at this, again, the head of a pin, this is actually an amoeba. And maybe the light isn't quite bright or isn't quite dark enough in here, but you can see it just moving along. It's going that way. And you can see that it's decided that this bacteria and this bacteria are actually tasty enough to eat. There, it just snapped that one off the surface. And now it's going to snap that one off the surface right there. And so they do this all the time to eat them. But occasionally these organisms actually cause diseases in those organisms, or they may go into the organism and live in that organism and remain there, where they can either kill the amoeba, or they, as a disease organism like Legionella, they can re-emerge from there, having survived various other treatments, and cause problems for us. We've also heard about protozoa like Giardia and Cryptosporidium. These can persist in biofilms as well. and viruses are also concentrated in microbial biofilms. So I'm sure you've seen Giardia and Cryptosporidium, these little guys here. If we look around in biofilms with antibodies, we can find there's a little cyst. Here's the whole biofilm, and here you can see the cyst separated out quite nicely. So that little Cryptosporidium cyst is sitting there in the biofilm waiting for a sloughing event to re-emerge it, for it to emerge into the overlying water. So biofilms are resistant to biocides. Monitoring is difficult. This is a major area in terms of what's happening inside of pipelines, what's happening inside of wells. And you get release of organisms, including pathogens, into the bulk fluid, which is a major concern. And the major challenges really in terms of a lot of aspects of drinking water, particularly upstream of treatment, is really this set and forget mentality. I can't tell you the last time I actually had my well checked, but there is that tendency to look at them from that perspective. There's also a very reactive nature to maintenance. I mean, I'm sure most people had not seen pigging of pipelines until it was done in Walkerton because they were concerned that they should clean out these pipes. So it tends to be very reactive. Monitoring is a problem. System design is a problem, and there's quite a bit of room for materials research as well in terms of how do you design, in the case of water wells, how are they properly designed? We haven't really moved very far from a hole in the ground lined by something to keep it from collapsing. So there's quite a bit of room to do work there. Some of this is being carried out as part of the Sustainable Water Well Initiative of Agriculture and Agri-Food Canada with the PFRA and Environment Canada. And just acknowledge Environment Canada, Health Canada, NSERC, and other people for contributing to the research that allowed me to pick out a few things that I could share with you today and hopefully give you a little bit of an idea about biofilms and about how you might think about them and why you might actually be concerned that they could be developing in your well or within your pipeline system. So with that, hopefully there's time for a question or two.